The eDNA Bacterial Aquatic Pump Kit includes a filter with a smaller pore size, ideal for targeting bacteria. In this kit, you'll receive a pair of sterile nitrile gloves, a sampling bag for collecting water, silicone tubing for use with a peristaltic pump, a filter for capturing DNA, preservative solution and filter caps, a disinfectant wipe for decontaminating any reusable equipment, a specimen safety bag, and a data sheet for noting information about your sample. You will need to take into the field with you a smartphone or GPS for recording location data. Remember to always check and obtain any licenses or permits you need before going into the field. Scan the QR code on the kit bag for full sampling protocol and refer to our guidance on how to choose your sample locations, how many samples to take, and how to subsample to collect water that's representative of your chosen area. Before sampling, it's important to put on the gloves provided to minimise contamination. Once you've collected your sample of water, you will need to filter it through our enclosed filter to separate out the DNA. Note that the top of the white line on the sampling bag indicates the 5 litre mark. To do this, you will need a peristaltic pump. If you do not have your own, you can rent a vampire sampler such as this one from NatureMetrics. The battery will come fully charged, but if you're taking lots of samples, you may need to recharge batteries using the charger provided. Insert the silicone tubing into the peristaltic pump and ensure it is pumping towards the lure lock end of the tubing, as this is where the filter will attach. You may need to adjust the flow direction using the red button on the side of the pump. Place the other end of the tubing into the water and run the pump to prime the tubing with water. This ensures there are no air bubbles left in the tube. Attach the filter to the end of the tubing with a small twist until you feel a slight resistance. Begin running the pump, starting slowly and gently increasing speed. Note that sometimes the tubing can suction onto the sampling bag. To resolve this, simply pull the tubing away to continue filtering. Continue to run the pump until your desired volume of water has passed through the filter, or until the filter is clogged. You'll know when the filter is clogged when it becomes too difficult to push any more water through the filter. The pump may begin to change pitch or make a clicking noise when the filter is clogged and the pressure begins to build up. Measure the water passing through your filter by measuring into a measuring jug like this one. Once you have finished filtering, lift the end of the tubing out of the water and continue to run it so that air is passing through the filter instead, and run this for about 30 seconds. Here you have your eDNA sample. To preserve your sample, take the small syringe from the grip seal bag, uncap it, and attach it to the filter with a small twist. Turn the syringe so that the filter is pointing upwards and slowly push the plunger so that the preservative solution fills the filter capsule. When you notice a bubble of solution coming out the top of the filter, take one of the filter caps and cap the top end. Then flip the filter upside down and push the plunger all the way in. It helps to give the filter a little shake to ensure the preservative is evenly distributed. Remove the syringe from the filter and cap the other end. Don't worry if some of the solution foams or spills over, this is normal. Place your cap filter into the specimen safety bag provided and seal. Fill out the datasheet with information about your individual sample, including a unique sample ID, which needs to be different from any other samples you may have taken. Place both your sample and your datasheet into the kit bag. Your sample is now ready to send to the lab for analysis. Once all of your samples have been collected, you must also fill in the provided electronic sample manifest. To return your samples, we recommend reusing the cardboard box that your kits were delivered in. As well as your samples, you may also return any waste plastic to us for recycling. Just ensure that it is washed, dried and sealed in a bag to keep it separate from your samples. Pack out any spare space with paper or packaging material. Securely seal the box and post it to the address displayed below using tracked postage. If you're sending samples from outside the UK, contact our logistics team who will arrange a courier service for you.